and she's a graduate student in our department, biological sciences. And she's doing a study of a food web to see where toxins are going, and also in food webs that are pretty contrasting, and ultimately interested in, are they getting to the top of things that we care about, more than maybe phytoplankton or zooplankton, where we should, obviously. But things like the top predators, in this case, it would be loons and fish and that sort of thing. So she's going to be talking about some pretty interesting systems she's trying to approach to actually track the cyanotoxins and see where they're going.
Great. Thank you for that introduction, Jim. So as Jim said, I am studying food webs. And the reason that I'm interested in food webs is because of this idea of biomagnification, where toxins become more concentrated as they move up the food web, as the trophic position increases. And that means that even if toxin concentrations in the environment are really low, top predators, those that are feeding high in the food chain, could still be exposed to high toxin concentrations. And because of the negative health effects associated with many toxins, we want to be able to predict that exposure so that we can then reduce it. And one way that we can do this is by determining how the toxin behaves in the food web. Is it becoming more concentrated as it moves up? Is it becoming less concentrated? And then also what these species interactions are. How are species, who's eating whom? How are they interacting with one another? And the toxin that I'm interested in is BMAA. It's a neurotoxin produced by cyanobacteria. And BMAA has been shown to biomagnify in terrestrial systems. And so kind of the classic example came from some work that Paul Allen Cox did back in 2003. And this was on the island of Guam. And they found that BMAA did biomagnify, and that was linked or suggested as a potential cause of a high incidence of neurodegenerative diseases in the native Chamorro people of Guam. But when we look in aquatic systems, evidence for biomagnification is pretty limited, and it's actually pretty inconsistent. And so we're not seeing that same pattern. And so I want to know, how is BMAA behaving in an aquatic food web? And I really think about this question in three parts. First of all, where it's coming from, what potential producers of BMAA are present in the lake, and are they actually producing BMAA? Where it's going, is it biomagnifying? Is it concentrating at a particular place in the food web? And then how does it get there? What are the kind of trophic pathways that led to that accumulation in that particular spot? And this last question is really getting at how different trophic lakes that have different trophic structures are affecting the patterns of accumulation. For example, we might think that a lake that has fish, the BMAA will accumulate differently than it would in a lake that doesn't have any fish. So I'm going to bypass all of the methodology. I used a lot of methods that have already been talked about by various people, and just kind of jump into some of the things that we found. So one of the first things that we noticed is that there's a lot of potential producers of BMAA in the lakes that I studied. All of the lakes that I studied are in Northwest Wyoming in the Yellowstone National Park area. But when we looked at the actual biomass of the cyanobacteria, which we measured based on fluorometric PC measurements of the phycocyanin, we found that the biomass was pretty low. We also found that the concentrations in the water, concentrations of BMAA in the water were also pretty low. And so our environmental kind of background level, not particularly high. But this wasn't that surprising to us. As I said, these lakes are in Yellowstone National Park and the surrounding area. And so they are very oligotrophic. They're, tend to be clear. I've worked on these lakes for the past couple of years, gone out to them almost weekly throughout the summer, and never seen a big bloom event. So like I said, this wasn't particularly surprising. But even when we don't see those blooms, we know from testing blood samples of balloons that they are accumulating a relatively high concentration of BMAA, sometimes up to 200 times what we were seeing in these background levels. So these kind of bloom-forming species, they're present. They are likely producing some toxins, but they're still not present in very high concentrations. This could be kind of the beginning of the evidence for biomagnification, but we do also want to consider what non-bloom-forming species might be present in the lake, what are not coming up in our regular counts. And so we measured PE, it's a pigment called phycoerythrin, which Nancy mentioned is often associated with Cyanobacteria. And the first thing I'll say about this is that we don't really have a good idea of what level would be high for a Wyoming lake. So we don't have the best context for this yet, but compared to some lakes that we've sampled in New Hampshire, some of the clearer lakes were closer to about 20 RFUs. This was pretty similar to a bloom sample that we collected from York Pond this year. So again, we don't have the best context for Wyoming lakes, but we think that this could be relatively high, which could suggest an abundance of Cyanobacteria. And that could be kind of our source of BMAA in these lakes. We also found that some of the lakes do have benthic mats of Cyanobacteria. And so the next step in that is just to kind of test the actual BMAA concentration. I've been working on the identification side of things so far, but now I need to overlay that with BMAA to see how these non-bloom forming species might be contributing to the overall BMAA concentration within the lake. When we think about where it goes, we do know that it is accumulating in the top predator in the loon. But like I said, we really are more interested in where it's actually concentrating. And so I went out over the summer to these lakes and sampled the most abundant species of plankton, plants, insects, frogs, fish, and then common loons. And basically what we want to do is to assign each of these organisms a trophic position. And we do this using stable isotope analysis and based on this formula. So it basically takes a stable isotope value for nitrogen. It measures the concentration in the consumer relative to a baseline value to calculate the trophic position. And so each individual will have its unique trophic position. And that way we can test, we can overlay that with the BMAA data so we can determine how BMAA concentrations change with changes in trophic position. And if BMAA does actually biomagnify, we would expect a relationship to look something like this, where as the trophic position increases, that BMAA concentration is rapidly increasing. But this doesn't really tell us much about how the species are actually interacting. It tells us how the toxin itself is behaving. But to show you an example of why those species interactions might be important, I'm going to show you two different food webs. In both cases, the primary producer is Cyanobacteria. Zooplankton would be our primary consumer. But in one case, the zooplankton are eaten by insects, which are then eaten by frogs. In the other case, the zooplankton are eaten by planktivorous fish, which are then eaten by piscivorous fish. And we would kind of expect, just based on a trophic position calculation, that the frog and the piscivorous fish would be feeding at roughly the same trophic position, right? They're kind of occupying the same level in the food web. But we also know that frogs tend to be hanging out in kind of shallow water areas, whereas these big fish are generally going to be in the deeper water areas of the lakes. And I know from the work that I've been doing with the plankton identification that the cyanobacteria community in the shallow area is really different than the cyanobacteria community in the deep water area. And so we would expect that even within a single lake, the toxin concentration, this kind of baseline level, could be very different. And so we not only need to know, is it accumulating at these top levels, but also the pathway that it's actually taking to get there, so that we can properly identify how the toxin is concentrating based on the correct baseline level. So I'm currently in the middle of this. I don't really have any coherent results to present to you guys today, but just to kind of give you an idea of where I'm at in this process and what's coming up for me, I'm going to be continuing plankton identification and counts. There's one type of plankton and one type of potentially cyanobacteria that we have not been able to identify yet, and it is one of the most dominant species in a couple of our lakes. So if any of you plankton identification experts want to give me a hand, I would be much appreciative of that. But then we're also continuing the ELISAs, the toxin analysis on a bunch of different samples, and I'm getting ready to run the stable isotope analysis so that we can kind of identify those trophic interactions, those trophic positions, and then overlay that with the toxin data. I have a ton of people to thank, but with that, I'll take any questions.